Good afternoon, students. And today we will be discussing the chapter sexual reproduction in the flowering plants. And till our last class, we have completed the first chapter, reproduction. In that chapter, as I already told you, that you will have a very brief account of the terms, but the elaboration, or we can say in that chapter you have the terms, but the elaboration of the sexual reproduction in the flowering plants, the human reproduction, will be followed in chapter 2 and chapter 3 respectively. So today we will be starting this new chapter, which is the sexual reproduction in the flowering plants. So now let us begin with the chapter sexual reproduction in the flowering plants. The first most important thing, whenever we talk about the sexual reproduction in the flowering plants, students, which part comes in our mind? Whenever we talk about the sexual reproduction, we are not talking about the vegetative reproduction in the plants. Whenever we are talking about the sexual reproduction in the plants, which part comes in our mind? Very good. Very quick answer, Pragya. Flower. Flowers. Yes, correct. All the students have given the correct response. Flower. Definitely correct. Flower. Correct. So all flowering plants, the angiosperm, they will show the sexual reproduction because they bear flower. So if someone asks you what is the site in the plant for the sexual reproduction, definitely the answer will be the flowers. Okay. Now, first of all, before going into the site of the reproduction, or whenever we will be doing the site of the reproduction, we have to always remember that we have to study about the parts of the flower. That is, typically, if we will say, the flower has two main parts. One is called the androecium and the other is called gynoecium. Okay, one is the androecium and the other is a gynoecium. Student, from the name androecium, can you tell me whether it is a male reproductive part or the female reproductive part? Androecium is the male reproductive part or the female reproductive part? Okay, very good, male. So obviously the next becomes the opposite. Gynoecium is the female reproductive part. Correct. Very correct. Yes. Now, if students, we are to discuss about the parts of the flower, first of all, the part which will attract the insect, definitely the bright colored petals. Okay. And we can see over here in this diagram, the androecium, though the parts, the subpart name we will come later on, but now we are, yes, see, this is the androecium. This is the androecium and this part is the gynoecium. Though there is structural detail now we have to do in our subsequent slides. Just a moment. First, students, we will be coming to the discussion of androecium. Androecium, definitely, it is a male reproductive part and it consists of whole of stamens. Their number and the length are variable in the different species. If you can remember, we were saying that the stamens are arranged in class 11. We have studied the terms monodalphous, didalphous. Okay, so whether they are arranged in one or in groups. So if we are talking about the stamen, first of all, if we are looking to the different parts of the flower, we can see stigma, style, and the ovary. this part, the stigma style and the ovary. This makes the female reproductive part, while the stamen and the filament, this is the filament where I have indicated as F. This makes your male reproductive part, that is the androecium. Now student, parallelly in this diagram, we can also see the structural detail of the stamen is also highlighted over here. The structural detail of the stamen is also highlighted over here. All of you look to this figure, 1 minute. If we are talking about the stamen, which is the male reproductive part, the stamen consists of the filament and the anther. Now, one very important thing regarding just a moment. Okay, so the stamen consists of the filament and the anther. Now let us discuss one by one. As you can see, student, in the diagram, the filament is a long and slender stalk. Its proximal end is attached to the thalamus or the petal of the flower. Here in this diagram, you can localize, or you can see over here that this part is the filament. This part is the filament. Okay, here also this part is the filament. Remember student, this diagram of the typical angiospermic flower is given in the NCERT also, which I will be providing you through the notes, which you all have to draw also later on. So this is the filament. This part becomes the filament. Now the major part is the anther. Now, first of all, student, remember in anther, the most, or we can say in board examination, many probable questions can be framed from the structure of the anther. First of all, look to this diagram. See how we have to move into the space. This is in total the stamen, as I told you. This part was your filament, which I'm indicating as F, and this part is the anther. Now the anther in the sectional view, you can see in this image. The anther in the sectional view. Now see how what is the structural peculiarity of an anther? It is bilobed. Okay, bilobe means, suppose if I'm drawing, let me first clear the drawing and then I'm redrawing this with a different color. See, if I'm saying this, I'm drawing this anther, two lobes. That means this is one and this is the second one. Got it? So if you are saying how many lobes are there in an anther, it is a bilobe structure. Is it clear to all? Is this clear to all? The point regarding the lobes, how many lobes are there? How many lobes are there? Is it clear to all? Okay. Okay, yes, you have given me the remark. Yes. Okay, that means it is a bilobed structure. Now, the second point, see, each lobe has two theca or dithecus. What does this mean? Now, for this, I'm going or moving to my next slide. See, students, when an anther is, a transverse section of the anther is done, you all focus over here. Okay, so these are the two lobes. Okay, lobe 1 and the lobe 2, and in each lobe you have two microsporangia. So this structure, the pollen sac, which you can see, this which I am rounding it off, this structure is the microsporangia or the pollen sac. So how many total pollen sac or the microsporangia present in an anther? For this I will unmute some of the students. Yes, Anjali. Anjali, unmute yourself. Yes, Anjali, unmute yourself. Yes, Anjali,
Yes,
how
sir.
many microsporangia is present in one anther?
4.
4. How you are saying 4? You are correct, but my next question is how you are saying 4. If I say how you are justifying this, though by the image you are justifying this, very correct, but you have to be more specific.
Because each lobe has 2 cup-like a structure called diathecus.
Very
Then we
good.
have 2 lobes that
Yes.
is called
Yes.
tetrasparangiate.
Very well explained. First of all, we have to say that we all know that anther is a bilobe, and in each lobe, 2 pollen sac is there. So 2 into 2, that becomes your 4 microsporangia. See, student, why we are going step by step? Because you will see, first of all, we have to do the transverse section of the anther. And already I told you in chapter 1, where we have done the reproduction and overview. As I told you, it was very superficial chapter where only terms were there. And then I said that in detail of discussion, this part will come when we will be doing the transverse section or the different parts. So an anther has 4 microsporangia. And now we will be studying about this developmental process, that how this microsporangia develops into the pollen sac. So these microsporangia develop to pollen sac and they extend longitudinally all through the length of an anther and are packed with pollen grains. So if you will see, student, the bottom diagram, if you will see, this is the pollen sac open, or the microsporangia, we can say, busted out, and you can see the pollen grains are present over here. The pollen grains are present over here. Did you got it? Is this part is clear to all the students till this? Okay, fine. Okay. Yes, I've seen your feedback. Okay. Now let me go to the next part. Now students, see, this is a very, very important. Every year in the board examination, we have seen the structure of the microsporangium always comes. See, once again, I'm telling you how we have or how we got ourselves introduced in this matter. First, we did anther overall, then the transverse section of the anther, where we have seen 4 microsporangia, and now each microsporangia. So in this diagram, which I have shared with you all, in this very first diagram, you can very well see that this is a pollen, this is a anther where 4 microsporangia are there, 1, 2, 3, 4, and the anther sac with the pollen filament. Now you see in this diagram, if I'm saying, we are showing the structure of the microsporangia. 1 microsporangia is now highlighted over here. Got it? How we are moving? Because structure of the microsporangia, we have to do very minutely and in details. Are you getting, student, how we are getting into the topic? As I told you, student, that this chapter will be a detailed account of the sexual reproduction in the flowering plant. So a typical microsporangia has 4 walls. Now see, I'm just zooming this image. It has 4 walls. First, see, what are the 4 walls? The outermost is the epidermis. After the epidermis is endothecium. After endothecium is the middle layer, and then the inside one is called tapetum. So remember student one thing, first of all, we have to say that it is made up of how many? It is made up of how many layers? It is made up of 4 layers from outside to inside: epidermis, endothelium, middle layer, the tapetum. Okay, now one by one regarding the different layers we will be discussing in detail. Yes, just a moment. See, out of these 3 layers, the outer 3 layer, that is epidermis, endothelium, and the middle layer, it gives protection and help in dehiscence, that is the bursting of the anther to release the pollen. But we will also study what the tapetum is going to do now. See what the tapetum is going to do. The tapetum, which is an innermost layer, it nourishes the developing pollen grain. Cells of the tapetum contain dense cytoplasm and generally have more than one nucleus. In young anther, each microsporangium has sporogenous tissue at the center. It consists of compactly arranged homogeneous diploid cell. Now here this new term has come to you all. Let me explain to you. As I told you, student, the tapetum is the innermost layer, and the outermost layer where the epidermis, endothelium, and the middle layer. This tapetum, which is centrally located, it contains the diploid microspore mother cell. It is a diploid. This is also called as MMC. That is a microspore mother cell. This is a microspore mother cell, MMC. Now we will also study during the process of the microsporogenesis that what is the function of this MMC, that is a microspore mother cell, what divisions or what differentiation it goes into account, and what are the subsequent developmental history. Students, is this clear to all? Regarding the structure, till now the structure we have discussed, is it clear to all? Is it clear to all the students? Okay, Srishti, once again, if we are seeing the cross section of a typical microsporangia, it consists of the 4 layers. Epidermis is the outer one, followed by endothelium, then the middle layer, and the inside one is your tapetum. The epidermis, endothelium, and the middle layer, they are protective in function. On the other hand, the tapetum is providing nourishment to the developing pollen grain, while it also contains a dense sporogenous tissue, which is called, or ultimately we will be saying that this structure is a microspore mother cell. Why it is called microspore mother cell? Because from microspore mother cell, microspores are, which is a unit of, or we can say where we will see microsporogenesis, how the microspores are formed. From MMC, the microspore mother cell, it gives rise to the microspores. Is it okay, Srishti, as you asked to repeat once again? Okay, very good. Okay, now anyone who can explain to me the structure of the microsporangia, please raise your hand quickly. So let me interact with you all also. Any student who can very well, or who can definitely raise your hand so that I can unmute you. Okay, Pragya. Pragya, you unmute yourself. I've given you the option. Pragya, unmute yourself.
Yes, sir.
Yes, Pragya. Could you brief up the structure of the microsporangia, which we have done just now?
Sir, am I supposed to tell you the 4 layers of the microsporangium which we did right now?
Yes, yes,
The
definitely.
epidermis and I just don't know why I forgot the 3rd layer, and the 4th one is tapetum.
Okay, you can see the image
Epidermis.
also, the layer, the name of the 3rd layer, that is a middle layer. Okay.
Sir, it's not clear in this slide, the name actually.
Yes. So what is the function of the outer 3 layer, epidermis, endothelium, and the middle layer?
All the 3 provide protection. All
Okay.
the 3 layers and provide nourishment to the developing pollen grains.
Okay, okay, well said, Pragya. You can mute yourself. Yes, Amrita. Amrita, you can unmute yourself, Amrita.
Yes, sir. Yes,
Amrita, good. Can you explain, Amrita, the structure of the microsporangia which we have discussed just now?
Yes, sir.
Okay, go ahead. I think you got again. Okay, I'm unmuting you. You can again unmute yourself, Amrita. You got.
Yes, sir.
Yes, Amrita, go ahead.
Microsporangia is made up of 4 layers: endothelium, middle layer, and tapetum. The first 3 layers, endothelium, endothelium, and middle layer help in protection, and last layer, tapetum, help provide nourish to develop pollen grain.
Okay. Okay.
Yes.
Well explained. Okay, Sakshi also has raised her hand. Okay, Sakshi, just a moment. Let me give you the option to unmute yourself. Yes, Sakshi. Sakshi Singh. Yes, you can unmute yourself, Sakshi.
Yes, sir.
Yes, Sakshi, want to share the same answer? Please go ahead. Briefly discuss about the structure of the microsporangium.
There are 4 layers of microsporangia.
Okay.
First, epidermis, second, endothelium, third, middle layer, or fourth, tapetum. The first 3 layers protect the microsporangium, and the
Correct.
tapetum is the innermost layer which provide nourishment to the pollen grains.
Okay, very good. Okay, Sakshi, you can mute yourself. Remember, students, the structure of microsporangium is a very important question which appear as a 3 marks question in the board examination always. Okay, so this part is very, very important. Okay, now student, we will be moving to our next topic. Now, the major portion of this lesson, and whenever we are talking about the male reproductive part, that how the microspores are formed. Student, before going, this is a schematic representation of the Dithecus anther. In the Dithecus anther, where you can see 4 the bilobed structure, and then altogether 4 microsporangia are there. This is a typical schematic diagram. This is a pollen sac, epidermis, fibrous layer, tapetum, and the pollen. And within the pollen, within the tapetum, the inside of the tapetum, you can see, as I told you, microspore mother cell, which is also known as PMC. That is a pollen mother cell. This pollen mother cell will undergo meiosis to form the tetrad of pollen, which gets separate. But how it happens, that we have to discuss. So as the anther develops, each sporogenous cell, the microspore or the pollen mother cell, undergo meiotic division. Students, who can quickly raise your hand to say that why it is undergoing meiotic division? Why the PMC, pollen mother cell, or the microspore mother cell. Pragya raised her hand at first. Okay, let me allow her to unmute herself. Just a moment, let me come to your. Yes, Pragya, you can unmute yourself. Why the PMC or the MMC? Huh.
Yes, the pollen mother cell or the microspore mother cell undergoes meiosis because it is diploid in nature and gametes are supposed to be haploid, so it undergoes meiosis or reductional division to produce haploid gametes or like
Very good.
grains.
Well explained. That is definitely the PMC, the pollen mother cell or the microspore mother cell is diploid, and for the gametes, for the fertilization, it has to be converted into the haploid. So it is undergoing meiotic division to form 4 microspore tetrad. Remember, student, this is very important for point. It undergoes meiotic division to form the microspore tetrad. That is a microspore arranged in a cluster of the 4 cells. Now, the formation of the microspore from a pollen mother cell through meiosis, it is called microsporogenesis. Student, can you recall one very important or the similar events which we have to say when we will be doing the human reproduction? If I'm saying microsporogenesis, can you recall that with which similar events we can connect it with the human reproduction? With which similar event we can connect it with the human reproduction? Very good, very good. Prashant, you have to be a bit specific. As you said, gametogenesis. Correct, Pragya, it's spermatogenesis, the process of the formation of the male gamete. And here the microsporogenesis, the process of the formation of the male gamete in terms of the plant. So now, as I told you, student, chapter 3, when we will be doing, we have to discuss in detail the sexual reproduction in the human being. And now when we are doing this part, with reference to the plant, we have to be specific. So the formation of the microspore, which is definitely the male gamete from the PMC, that is a pollen mother cell, through meiosis is called microsporogenesis. And what are the events which will be taking place now? We have to go to the detail of this also. See what happens. As the anther matures and dehydrates, the microspore dissociate from each other and develop into pollen grain. Each microsporangium contains thousands of pollen grain, and they are released with the dehiscence of the anther. Now, student, this word, the dehiscence of the anther, and you can see, let me go back to one more image. In the labeling, in the diagram, one labeling was there, line of dehiscence. Okay, you can see in this image, there is a labeling over here, which is called the line of dehiscence. That means the released pollen will follow this line for the dehiscence. And that is why it is called the line of dehiscence. Okay. Just a moment. Next. So, now remember, students, whenever we will be talking about the male gamete, we have to do in detail the reference of the pollen grain also. The male gametophyte, which is the pollen grain, it is generally spherical, and cytoplasm is surrounded by the plasma membrane, and the pollen grain has 2 layers, the outer exine and the inner intine. Let me zoom in more. You can see in this diagram, The pollen grain is, first we will be talking about the two layers. This one is the intine and the outer one is the exine. Remember student, this is very important. Okay, why? Because whenever we are talking about pollen, what makes the pollen so tough? Okay, that we have to discuss in detail. What is present in the exine which makes the pollen so tough? Because intine is a thin layer while the exine is the tough one. Yes. See, why the exine is so tough? Remember student, this is a very important topic, that why exine is so tough. Actually, the exine, which is the outer layer of the pollen grain, it is made up of sporopollenin. Remember, this is a very, very important substance because it is highly resistant organic material and it can withstand a high temperature and strong acids and alkali. And that is why the pollen grains are preserved as fossil, because the sporopollenin is not being digested or disintegrated by any of the material. And this question becomes a very important objective question, that why the outermost layer of the pollen grain, which is the exine, is so hard. So the outermost layer of the exine, which is hard, the outermost layer of the exine is hard due to the presence of the sporopollenin, which is a highly resistant organic material. Now see, this exine has pores, which is called germ pore. And there the sporopollenin is absent. Why the sporopollenin is absent? Because it has to function in the reproduction, and if the exine is present all over the parts, then the liberation of the male, or we can say the liberation of the germ cell, won't occur. So these are the germ pores through which the germ cells will go, or we can say the act as a channel for the germ cells. Okay, so this sporopollenin is the germ pores are the place where the sporopollenin is absent, and these pollen grains are definitely preserved as a fossil due to the presence of the sporopollenin. Till this clear to all of you? Till this, is it clear to all of you? Yes, I'm waiting for your responses. And student, the next layer which you can see, the next layer which we can see is the intine. The inner wall, it is thin and continuous, made up of cellulose and the pectin. Now, students, yes, till the pollen grains will be our today's topic of discussion. So student, today's assignment will be definitely this, till today, whatever we have discussed, the note part that I will provide you, which you have to fare it up. So that part will be done today. Okay.